It's our pleasure to meet you all here after yesterday's, for some of us, visited the field and some of us stay here. Hope you enjoy the visit and also have the really good rest last night. So today, the session will be led by Professor Dami, but prior to that, Scott will present some updates. So please welcome to Scott.
Thank you. I think some of this information will be quite useful for you when you go through the exercise of the risk assessment. And I think I personally am not going to have any involvement with the risk assessment other than being a potential resource if people have questions of me. But I do want to think it's be useful to be able to just let people know where the current state of the program is, and also key scientific pieces of information that will be relevant that has been published in the literature, but an opportunity for me to share with you. So the Eliminate Dengue program, as we call ourselves, is an international collaboration that sits across 5 countries at the moment. So not just Indonesia, but also in Australia, Brazil, Colombia, and Vietnam. In case you are not aware, while dengue is a huge problem here in Indonesia, it's a problem throughout the tropics of the world. So it's estimated that 3.9 billion people are at risk every year of getting dengue. That's about half the world's population. And it's estimated that the cost of dengue to governments is around $8.9 billion US every year. So it's causing a very large problem around the world. And when you look at it at a family level, most of the cost actually, or a large proportion of the cost, sits with families, not just with governments. And in many developing parts of the world, those costs are extremely high for people to be able to deal with. In the last year, there's been a global public health emergency declared from the World Health Organization for another virus, not dengue, but another virus called Zika. And Zika is a virus that's transmitted by the same mosquito, and so the ecology, the biology of the disease is very similar. Just in case you're not aware, this virus was first described from the Zika forest in the 1930s in Africa, and in the last, probably 18 months now, there's been a very explosive outbreak occurring in South America. Just within the last year, there have been almost 6,000 cases of microcephaly. These are babies that are being born deformed with improper brain development, and so small heads, and many of them either dying or needing long-term care as they grow. And so this is against a background of usually around 163 cases a year. So a huge spike in this very distinctive and recognizable condition. It's now appreciated that there's a much broader spectrum of neurological disease than just microcephaly occurring, and that this problem is looking like it's a very large problem around the world. Since it's been declared, the outbreaks were declared in Brazil, within the last 12 months, it's now spread to another 60 countries. And that's part of the rationale for why the World Health Organization has declared this an emergency. So the approach we're taking is using an intracellular bacteria called Wolbachia. So what is Wolbachia? So its taxonomy puts it closely related to other intracellular bacteria that are more commonly known, such as the Rickettsiaceae, so different Rickettsia, Anaplasma, Ehrlichia. But Wolbachia is only known to infect arthropods. It's not known to infect vertebrates. There's been no evidence reported of it being able to, and nor can it be introduced into vertebrates. It's originally taken, the strain that we're using in this study is being taken from the fruit fly, Drosophila melanogaster. This is a picture of it up here. It's very common around the world. And in nearly all parts of the world, you will recognize it in your kitchen maybe if you have some bananas that are a bit too ripe. And as they become very ripe, draws these flies in from the environment, and they're very characteristic by their little red eyes. So this fly, Drosophila, occurs around the world, and everywhere that Aedes aegypti occurs, Drosophila melanogaster occurs. And so in our study, we have taken this bacteria. I don't know if there is a pointer. We've taken this bacteria from this fly and put it into the mosquito so that it is vertically transmitted through the eggs. Many people say, well, that seems like a good thing to do. Why don't we just do that locally? Why did you bring your stuff from Australia to here? Why don't we just do it in Indonesia? And I think the piece that is not well understood is that this is the actual process. These are mosquito eggs here, and this is a very fine glass needle that we prepare in the laboratory. To actually do these transfers is very, very difficult. Thank you. So I think there are only 3 groups in the world that are able to do this reliably at the moment, and to develop the strains that we did, we had to inject thousands and thousands of embryos, and over the last 10 years we've been able to generate in my laboratory around 4 strains. So it's possible for people to redo the microinjection, but it's likely to add 4 to 5 to 10 years to the process of being able to move this intervention out. And so we don't recommend that. Instead, we recommend that you take the strain that's been developed and then repeatedly cross it to a local mosquito genotype to localize it to the local mosquito genotype of an area where you're wanting to introduce it, rather than having to re-establish it through microinjection. This is a phylogenetic tree of Wolbachia, and I just want to say a little bit about how Wolbachia is classified, the systematics of Wolbachia, if you like. So, these are all different strains of Wolbachia. This is an unrooted tree, but essentially if it was rooted, what you would see is that the whole Wolbachia group is monophyletic. But within this group, there are some different clades which are quite important. So this group down here on this tree is the W. Mel strain that we're using in the mosquito. And this is what's called the A supergroup of Wolbachia, and here is the B supergroup. There are a couple of supergroups up here called C and D, where the Wolbachia only infect nematodes, filarial nematodes that cause human disease. These Wolbachia are really quite unrelated to these Wolbachia. And at the moment, bacterial taxonomists are in the process of giving these Wolbachia a new name. So there's been no examples of where the Wolbachia that infect these filarial nematodes here are able to infect insects like the ones that we work on. So their biology and their genomic organization, their classification is quite different. So this is the key point here, that no Wolbachia strains from nematodes have ever been successfully transferred across to insects or vice versa. So when you are looking at the literature about Wolbachia, you can find papers about Wolbachia in nematodes, and it can be confusing because you might think they're the same as the Wolbachia in insects, but they're not. They're very different. And so I think in your thinking you need to be able to discriminate between these 2 groups of Wolbachia. Wolbachia is naturally very common. These are just examples of different invertebrates that naturally carry Wolbachia. So we estimate that up to 60% of all insect species have Wolbachia. And I don't know if there maybe is a taxonomist in the room here that can tell me how many species of insects there are on the planet. Last estimates I heard, that there were around to 8 million species. Can people hear me okay? Maybe I'll get rid of this. Can you hear me now? Okay. Okay, I have to do. So, around 60% of all insect species carry them. Maybe there's 5, maybe there's 8 million species of insects on the planet, probably less than 10 million. This means that this bacteria is incredibly common. If you were to just go outside of the hotel here and collect species off the plants of insects, just collect a random collection of insects, you could expect that around 60% of them would naturally have Wolbachia. So very common in the environment already. It's also common naturally in insects that bite humans, including a number of mosquitoes. They're nuisance mosquitoes. They're not vectors of disease agents to humans, but they still carry Wolbachia. I'm not really. My cultural background means that I'm not really that interested in eating insects, but many parts of the world people do eat insects, and many of these insects carry Wolbachia as well. And so people are coming into contact with Wolbachia regularly. They're ingesting Wolbachia and Wolbachia byproducts regularly. Also, there are many, for the entomologists in the audience, many biocontrol programs have been implemented around the world, probably in Indonesia as well, where insects are deliberately released into agricultural control programs. And many of those biocontrol releases are with insects that contain Wolbachia, but nobody ever bothers to look for the Wolbachia. And so there have been many natural experiments of introductions of Wolbachia in agricultural biocontrol programs around the world. So how is it that Wolbachia is able to be so successful? And what's even more surprising about Wolbachia is that it's not infectious. So if I was an insect and I had Wolbachia, I wouldn't transmit it. If we were all insects, I wouldn't be able to transmit it to you. It's only vertically inherited. I'm a male, and because I'm a male, the Wolbachia would not be transmitted by me. It's only transmitted through females and through the eggs they lay. So what's really unusual, though, about this is that if a Wolbachia infects a male insect, that infection modifies the sperm of that male, so that if this male with Wolbachia was to mate to a female that didn't have it, her eggs wouldn't hatch. And this is a very well-described phenomenon known as cytoplasmic incompatibility. If you had two insects that had Wolbachia, male and female, this female's eggs would hatch and all of her babies would all have Wolbachia. Or if the female had Wolbachia and the male didn't, it's the same outcome. She produces eggs that have Wolbachia and they all hatch. So this is a very clever, sneaky way that Wolbachia has to be able to spread itself into insect populations without needing to be infectious, just being vertically transmitted. And it does that by preventing females that don't transmit Wolbachia from reproducing. And it's this mechanism that allows Wolbachia to be able to sustain itself in an insect population. And I'll show you some examples in mosquitoes where Wolbachia is sustaining itself for many years after it's been introduced through this mechanism of cytoplasmic incompatibility. So it's very key to the biology of Wolbachia. Let's just talk a little bit about human safety and what do we know about Wolbachia and human safety. So Wolbachia was first described or found in 1924, so it's been known for many, many years. It was discovered or described in the USA, in Boston, on the campus of Harvard University in the School of Public Health. And it was in a mosquito population that people were studying on the campus, in Culex pipiens mosquitoes. And when they first found it, they got quite excited because they thought it might be a new pathogen, a new disease that they had discovered. And so they did the critical test of the time, which was they took preparations of mosquito ovaries that contained the Wolbachia, and they put them into chick embryos and also into mouse, and injected them into mouse brains, looking for any signs of pathology. And they were not able to see any signs of any evidence that would suggest that it was a pathogen, Wolbachia. And they became quite uninterested in it then. It just became like a boring insect symbiont, and it didn't look like it was pathogenic, and so people stopped studying it for many years. There has been some evidence, a number of papers published that are cited here, that it is possible to grow Wolbachia in mammalian cell lines in the laboratory. So these are like artificial in vitro systems of growing cells in plastic plates in media. And you're able to force Wolbachia into these cells and then select for the cells that are competent for infection. And so it's possible to form these artificial infections in mammalian cells in the laboratory, but there's never any evidence been demonstrated to show that you can actually get a Wolbachia infection in a living organism outside of this artificial in vitro system. So one of the key safety issues is, can Wolbachia be transferred to humans? We've done a range of work around this. So if the mosquitoes that we're wanting to release have Wolbachia, could they be transferred into people? So we've been able to look for the presence of Wolbachia in mosquito saliva and salivary glands, and we've been able to do that by looking at amplification of a well-known Wolbachia gene called WSP, and also mosquito A pyrase as a control. We're able to pick up the mosquito genes out of saliva, but we cannot pick up the Wolbachia gene in the saliva, indicating that Wolbachia is not present in the saliva. We can actually even collect expectorates from mosquitoes of their saliva and then PCR screen them, and we have not been able to detect any evidence of Wolbachia in the saliva, which is what would be needed to get transfer to humans. And this work has been published. We've also done a range of experimental studies where in my laboratory we maintain the mosquitoes with Wolbachia on human volunteers. We need human blood for the mosquitoes to grow, and so we use people to provide that blood. And so we have in my laboratory people who have been exposed to very high levels of biting for many years. So we would expect these people would be good people to look in for any possible transfer. So we've looked at sera collected from human volunteers that were either blood feeders or non-blood feeders, and then we've looked to see whether there's any, using Western blots and ELISAs, looked to see whether these people had produced any antibodies that target Wolbachia antigens. And what you're looking at here in this data between blood feeders and non-blood feeders, here when we look at antibodies against mosquito proteins, so in the saliva, we can see that the human volunteers have a definite antibody response against mosquito saliva, but not against Wolbachia from Aedes aegypti cells or taken from Drosophila adults. And so our conclusion is that Wolbachia antigens are not injected into humans during blood meals. So this supports what I was saying in the previous slide. We've also had the opportunity now to be able to directly test this. So we've been releasing mosquitoes now in 5 countries, and in our work we've done in Vietnam, we've been working with a community, which I'll tell you a little bit more later, where mosquitoes containing WML have been released and put out there. And then we've been able to bleed the people on that community before we released Wolbachia and then afterwards. So we have paired sera collections. And what we'd be interested in trying to address with those collections is there a rise in antibodies against Wolbachia after exposure to the mosquitoes that have been released on the island. So this is the experimental design. we took 145 people in paired samples, 2014 before we released mosquitoes, and then 1 year after releasing mosquitoes. We put them into an ELISA test, and that we wanted to look at the difference between the signal before and after. And for us to be able to show a positive response to Wolbachia, we wanted to see an increase in our ELISAs that was greater than 3 standard deviations. And this shows you the data from the 145 people on the island. This was what the cutoff value would need to be to show a positive antibody response to Wolbachia. And we see that there are shifts between people in the paired samples before and after, but that we don't see any response of the magnitude required to suggest that there has been any reaction to Wolbachia. So at the moment, we're doing field trials in Vietnam, Indonesia, Colombia, Brazil, and Australia. And I'd like to take you through some of those locations and what we've been doing. I think what's quite interesting from a human safety point of view is that now we're in a point where we have released Wolbachia mosquitoes over close to 200,000 people now over the last 5 years. And in particular, you know, our 17 field sites in Cairns started in 2011, so that we've had people living in communities continuously exposed to mosquitoes with Wolbachia, and there's been no adverse consequences been described for any of the human populations in these countries where we're working. So no safety signals at all in any of the work that's been done. What about the idea of Wolbachia being able to be transferred into non-target organisms, not humans, but other organisms in the environment, honeybees, ants, whatever you might like to choose? A number of experiments have been done and published. This is an example of one that was published in 2010, where mosquitoes with Wolbachia were fed to predators, in this case spiders, fish, copepods, and other mosquitoes that live together, like Aedes albopictus that lives often next to Aedes aegypti. And then we've used PCR to detect the Wolbachia DNA. So we've looked in, these are the species that have been looked at: fish, copepods, the spiders, so another mosquito, Toxorhynchides. And in this published work, there was no ability to be able to detect Wolbachia DNA in the insects that had fed on the Wolbachia-infected mosquitoes, so that we weren't able to demonstrate any sense of horizontal transmission. We also looked in the environment. This is a field cage that was built in Cairns, Australia back in 2009 when this work was first being done. And we would, in these outdoor cages, maintain populations of mosquitoes. And then we had other organisms within these cages where we went and tested for the potential Wolbachia transfer into those organisms. So we examined soil, plants, millipedes, different organisms in this environment to see whether we could see any evidence of Wolbachia in these environmental samples. And using PCR-based techniques, we're not able to detect any Wolbachia DNA in these non-targets. Again, this work is published and available. So just moving away from safety and talking about the reason that we're so interested in trying to introduce Wolbachia into mosquitoes is this impact it has on disease. And so there's now a number of papers that have been published and are available in the literature that show that Wolbachia in mosquitoes blocks all serotypes of dengue viruses. It also blocks replication of yellow fever virus, West Nile virus, chikungunya, Zika, and surprisingly there are a number of papers published now showing impacts on filarial nematodes and also malaria parasites. So this effect is very broad-based that we've been observing. This is all published. Based on this published evidence, we can make predictions about what other pathogens we would expect might be blocked, And I think it's fair to say that we would be very confident that we should be able to see blocking with Japanese encephalitis, although that experiment hasn't been done. Based on what we see with, for example, with chikungunya, we would expect other alpha viruses like Semliki forest virus or Venezuelan equine encephalitis to be blocked, and also other species of Plasmodium. So if I just focus in on dengue, which is our main target, the work that's been done on dengue, and a lot of work has been done in Ho Chi Minh City in Vietnam, where patients coming into a clinic with dengue have blood taken from them and then fed to mosquitoes with and without Wolbachia, with the idea of trying to understand what degree of blocking. And that data has then been put into various simulation models. This was an important paper that was published in Science Translational Medicine in 2015, and it shows the data from feeds on 62 patients in Vietnam, and this is the predicted reduction in R0 for each of the dengue serotypes that was observed in those experiments. So just to remember, R0 is the number of new cases that any one case should generate. So if I have dengue, then if I was to infect, say, 5 other people, then the R0 would be 5. And so we're looking at a 70 to 80% reduction in R0, and the view is that this number is conservative. What does a 70 to 80% reduction in R0 mean for disease in a community? And this is what's work that's been done by Neil Ferguson at Imperial College in London, and where he has looked at simulation models that go for 100 years tracking dengue transmission. And on this graph, we're looking at the reduction in dengue disease. And so very quickly, this scale is not linear, so 1, 2, 3, 4, 5, and then 10, 15, 20, and then 30, 40, 50. So you can see very quickly the reduction in disease rises to 100%. That means that there is no transmission of dengue. And then it continues until we see some small amount of transmission occurring once we get out to around 50 years after the introduction. And the reason for that return of some transmission is because the blocking effect of Wolbachia is not complete. It's a partial blocking effect. And we see that we get that bounce back, if that small bounce back occurs in R0 settings of around 5. It's very almost imperceptible at lower R0 values. An R0 of 5 is getting close to the R0 for measles transmission, which is a very high R0 and quite unusual for dengue. Most epidemiological settings would have R0s for dengue in the 1.5 to 4 range. And for dengues 2, 3, and 4, you don't see any of this bounce back. It looks like total elimination of transmission for 100 years in this simulation model. So the predicted impact is quite large. I started off mentioning Zika virus to you, and this just shows some recent data on the effect of Wolbachia on Zika virus. Really, this is with mosquitoes from Singapore that have Wolbachia, and they've been challenged with Zika. And I just think this, I won't concentrate on this graph so much, but this, because I think it's more important. This is where the salivary glands of the mosquitoes are, so this is a proxy for transmission. These are wild-type mosquitoes that don't have Wolbachia, and they all get infected with Zika virus very easily, as you would expect in their salivary glands, but in the presence of the WML Wolbachia strain, there's very little virus present in the salivary glands. And this data, this degree of blocking is around what we would see, or maybe a little bit stronger than what we would see for dengue. And so the predictions are that not only should you be able to get elimination of dengue transmission, but also of Zika. Sometimes I get asked the question, what about Aedes albopictus? Because Aedes albopictus naturally has Wolbachia, we know, but it's also known to be a vector of dengue. So why isn't Aedes albopictus, why aren't we seeing blocking? And actually we are. This is a paper that was published from Institut Pasteur in Paris in 2012. And in this experiment, they took Aedes albopictus and they removed the Wolbachia from it. And when they removed the Wolbachia from it, they did the vector competence experiment and showed that if you don't have Wolbachia in Aedes albopictus, that actually Aedes albopictus becomes a better vector. So it's more competent for dengue. So Wolbachia is having an effect, but it's not stopping transmission. So why in Aegypti does it stop transmission but not in Albopictus? And the reason we think is because the tissue distribution of the natural Wolbachia and Albopictus is restricted just to the ovaries of the insects, whereas the WML strain that we've put into Aedes aegypti infects many tissues of the insects, in particular the salivary glands and the fat body. And the fat body is a key organ in the insect for its immune response. And we think these tissues that are being infected are quite critical for Aedes aegypti, which makes it a much stronger blocker of dengue, the WML strain, than the natural strain that occurs in Aedes albopictus. So if you have questions about Aedes albopictus, I would really encourage you to read this paper. Because of the global health emergency around Zika virus, the World Health Organization made a formal declaration of a global health emergency this year and has been looking at new approaches that could be used for control of Zika. And there have been is an advisory group set up in WHO in Geneva called the Vector Control Advisory Group that brings experts together from around the world to provide advice on new vector control strategies. And in 18th of March this year, so just less than 2 months ago, or around 2 months ago, the WHO VCAG group reviewed the Wolbachia as well as other new interventions for dengue and Zika and made these statements. They looked at the evidence and indicated that Wolbachia reduces the mosquito's ability to transmit arboviruses. They didn't see that this was controversial, and that they accepted the evidence that Wolbachia reduces viral replication of dengue, chikungunya and Zika. And then they came out with recommendations and recommending to governments that pilot deployments, so this is an important word, these are not trials, these are actual deployments, pilot deployments under operational conditions and rigorous independent monitoring and evaluation take place. And they also recommended that current plans for randomised controlled trials, which will gather gold standard evidence on efficacy, continue. And that is actually what is planned for Jogjakarta in the next 3 years. This is the plan. We're planning to do a randomised controlled trial here in Jogja to test the method. So the World Health Organization is indicating that they think this should go ahead. So we've been working in 5 countries where we've been releasing mosquitoes to date. This just shows you the regulatory pathway that's occurred in each of those countries. There's no country that said no at this stage to releasing Wolbachia, but each one has had a different way of evaluating and thinking about it. So in Australia, we had independent risk analysis done through the CSIRO group that's here today working with you, and then ultimately approval from the Australian Pesticides and Veterinary Medicines Authority. And in each country, we've seen different approval processes. I think the most rigorous approval process has been in Brazil, where it's gone through numerous regulatory approval bodies before being given approval for release in Brazil. But part of what we do in our program is that we feel very strongly about that it's not sufficient just to have regulatory approval to proceed to release. So we have a very strong commitment to community engagement and having the community being supportive of what we're doing. In our early trials, particularly in Australia, we treated this almost like a medical research intervention where we wanted to obtain individual consent from people. And so we would have targeted community engagement, communication activities, door knocking, and we'd actually get people to sign consent forms for release of mosquitoes. So that worked quite well with our small initial releases, but we're now getting to the point of wanting to deploy Wolbachia over populations of 2 to 3 to 10 million people. And so individual consent models are no longer appropriate for us. So I just wanted to say too that in these early models, residents had the option to say no to releases. So it's possible for residents to opt out. Even if the government says it's okay, individuals are able to opt out of the experiment. But we can't scale that. So we've been moving throughout the program in different countries now to a new model of community engagement, which we're calling the public acceptance model. And it relies on a series of public participation principles or values, which I'll show you on the next slide. It's also key features are a community reference group, so there's a group from the community that oversees the community engagement to make sure that it's done at a high quality and what we say we will do actually happens. It's underpinned with quantitative social research that measures awareness and acceptability of the method. We have awareness raising and information sharing campaigns, and these are contextualized in different countries depending on the cultural environment. In Colombia, we do a lot of school plays. In Yogyakarta, I'm amazed to see this bicycle campaign that's going around at the moment, which I think is lovely. So each location does it slightly differently. And then the last piece that we see critical to this is that we have an issues management system so that people are able to complain, and when people complain, we're able to respond quickly to those complaints and address people's concerns. So I talked about the public participation principles. These are what we consider the key public participation principles, which we think are fundamentally important and not negotiable for any of the places where we do Wolbachia releases. And so firstly, the first principle is that we're respectful, so that issues raised by people are treated as valid. We don't ignore people who complain, and we consider everybody's opinion. Doesn't matter whether the opinion comes from a high-ranked individual or a low-ranked individual. We wish to consider all issues in the same way. We're inclusive, so we make an effort to include everybody within our engagement. So anybody with a potential interest in the project has access to our communications, and people are able to nominate their interest in being included, so that people can say we wish to receive information, and then we'll provide information. We're transparent. So being transparent means that we're clear, we're open in what we're doing, and so project information relevant to the community understanding is readily available, and it's kept up to date, so it's current. We're responsive, so that when we have requests or concerns, we make a commitment to be able to respond quickly to those concerns. And then finally, the key principle here is that we're honest. So we tell the truth. We're not trying to present our work to be something that it isn't. And so all of our communications are factual and cover the information of potential interest to people. And we're doing it in a way to make that information accessible. So in doing this program, the technology is actually very easy to implement. So in Australia at the moment, we're using these small little boxes in which we place water, some mosquito eggs, and here you can see mosquito eggs being produced here in Jogja, and we add some food, and we leave these in the environment where the mosquitoes will grow up and then emerge themselves over a 2-week period. We've also have done many experiments where we've released adults, which we grow in the lab and then release into the environment. And in some settings, adult release may be better than egg release, and in some settings, vice versa. So, let me take you through some of our field sites and what we've learnt so far. And I'd like to start in Australia, because this is the place where the first releases of mosquitoes took place in 2011. And this is a really key point I want to make about the Wolbachia intervention. So this is back in 2011 when we released mosquitoes, and these graphs are just showing the percentage of Wolbachia in the frequency of Wolbachia in the mosquito population in this location. 2 locations actually, suburbs called Yorkeys Knob and Gordonvale. This was our release period. We released mosquitoes in this experiment for 10 weeks, and then we stopped. And the Wolbachia levels were intermediate when we stopped, and then just by themselves they continued up to close to 100%. And then we monitored these mosquito populations, and we have been ongoing monitoring to see what happens to the Wolbachia. And the Wolbachia is staying, it bounces around a little bit, but is staying close to 100% in those mosquito populations without us doing anything. So as opposed to a vaccine, or as opposed to a source reduction campaign to remove mosquitoes, or an insecticide-based campaign, all of those programs, you have to have them ongoing. You have to sustain an ongoing program for the intervention to work. You have to either keep vaccinating children, you need to keep applying insecticides. In this approach, we believe this will be self-sustaining, so that once you have put in the intervention, it stays in by itself after that. So it doesn't need continual reapplication. So this is a very key point for both cost effectiveness. It means the cost effectiveness of an intervention like this will be very high because you're getting a continuous health return after you've deployed it. And it's also, I think, a very important point when you think about the sustainability of public health programs. It's often possible to get a donor to be able to put a significant amount of money in to start a program, and in this case the Tahir Family Foundation has invested heavily into this program. But it's very hard to maintain programs for many years, particularly when they are successful. As disease incidence comes down, it's very difficult to sustain high levels of investment, and that's seen throughout the world in all public health programs. The nature of this intervention is that the cost is front-loaded. So it just needs to be applied at the beginning, and then people like the Tahir Family Foundation can move to the next project they're interested in funding, whereas the benefits of this program should be felt for many years in the community. So in Australia, since those early experiments, we've moved into deploying into 17 suburbs over the last 5 years of Cairns, the northern tourist city in northern Australia. And we've found that we now have high levels of Wolbachia, which is the dark green, in nearly all these suburbs we're working in. And interestingly, none of these areas now have Wolbachia dengue transmission that's been recorded over the last 5 years. So no evidence of locally acquired dengue for people that are living in these areas. But that's not an efficacy trial, that's just observational data that we're gathering. This just shows you, we don't have anywhere near in Australia the level of dengue that you have in Indonesia or here in Yogyakarta, for example. But this is looking at dengue cases across Cairns, and these are looking at particular suburbs where Wolbachia has been released. And we're seeing after we get released that we're not seeing any dengue. We do see some little dots here, and what they represent is people that have acquired dengue outside of this suburb in another part of the city that's not protected, and then brought it in. So we haven't seen the key characteristic we'd be looking for local transmission is clustering of cases. So cases where we get multiple cases occurring in one location in a very short period of time, or in a very small geographic location. So it's quite common if you have multiple family members may get dengue in the one house within a 2-week period or something like that, as the mosquitoes are biting people within the house. But we don't see any evidence of that when we do see cases in these transmission areas, and so we're believing that they're not acquired locally. This is also showing you one of our release sites. It was where we released a few years ago in Cairns. This is the area here called Parramatta Park. It's a high-risk area for dengue in Cairns. So when there is a dengue outbreak, when we're thinking that we have transmission in northern Australia, this is an area that's usually sprayed with insecticide to protect residents before the outbreak really gets going. And so these red dots represent over the period between 1995 and 2012 the different locations of dengue cases within this area. And you can see the tight clustering of them in this area called Parramatta Park. And this is after the release period. We're looking at dengue cases again. So you can take a 1 kilometre boundary around this area and look at the proportion of cases that occur in this release area versus outside, and you can look at that proportion before and after transmission. It's an observational data measurement. This is what it looks like. So before release, 34% of the cases in that area came from within the release area. After release, only 4% of the cases came from. And these 4%, we believe, again, are people that have acquired dengue outside of the suburb because there's no clustering, and also from the travel histories that have been recorded from the people. So this is not an efficacy trial, it's observational data, but it's pointing in the right direction of what we would predict if the modelling is correct. We've since gone on to the city of Townsville in Australia. So this is a city of around 280,000 people, I believe. And we're in this city wanting to do large-scale deployments across the whole city. And we've been trialling this new public participation model of community engagement and wanting to drive our costs down. So in the last couple of years, we've deployed successfully over around 43 square kilometres. So that's an area around the size of Jogjakarta city. We've put Wolbachia in, so it's at high level across all these areas. But whereas Jogjakarta has around 500,000 people living in this area, in Townsville, Australia, there's only 50,000 people, so the population density is very low. And the cost for us to do that has been around US $16 per person. So that's a pretty large number, US $16 per person for an intervention. We can take that $16 and we can work out, how we spent that money. About 30% of it was on deployment, about 20% on monitoring, about a quarter of it was on community engagement, so a very high amount of money on community engagement, and then 25% was on other things like vehicles, administration, etc. So US $16 per person is not a very good number from the point of view of wanting to have an intervention, even though it's probably could be about the cost of, say, a dengue vaccine. We think we need to get that number much lower. And our goal is to get this number to be less than $1 US per person. And we think that we can achieve that quite fairly easily, actually. And one of the easiest ways that we can take this number from US $16 per person to a lower number is to put more people into the area. So if we were to take this cost and do this deployment the same way we did it in Townsville in Jogjakarta, because of the increased population density here, the cost immediately comes down per person to around $2.50 per person. And so what we would like is to be able to move that cost down to less than $1. And I think we're well on track, and some of the big deployments we're doing at the moment is working out ways to shave the cost down to deploy. This is just showing you dengue cases in Townsville. Every year for the last 15 years there's been dengue transmission occurring in the city. And then in the last 18 months there hasn't been, just one case, and that case was outside where Wolbachia had been released. So the public health people in Townsville are very excited about what they're seeing, but still not an efficacy trial. So currently we've deployed over this area here, which is what we call our phase 1 release. We've started releasing phase 2, which is a much bigger area, and we've already completed our deployments into these areas here. So we're expecting that we will complete this out with coverage over around 90,000 people by the end of this year. One thing that we've been doing quite differently in Townsville has been to use the community directly in the intervention, in the deployment of the intervention, and we've been particularly interested in using school children to do it. And so we've developed a school program which we trialled in 2015 in a school where we targeted 5 to 12-year-old children, and we had 105 of these children we called Wolbachia Warriors, and each of these children was tasked with taking home a kit to their house where they had to undertake 3 subsequent releases, and then of mosquitoes through their kit. They also had a calendar to mark significant events. There was some natural history experiments about describing the life cycle of the mosquito, and were able to record on their sheets observations around the release that was sent back to us. So it became a part of their science curriculum. Overall, we found this to be quite a successful method to be able to deploy. We're expanding the program this year into 4 more schools and going from 100 Wolbachia warriors up to 2,000 warriors. And so we think that through this mechanisms like this, we can help make the method more cost effective, but also get much more effective community engagement. We find it's using school children is a very effective way to engage adults through the adults working with the children on the project. And we find that to be more effective than trying to often engage adults directly. So it has a very strong community engagement benefit as well as a scientific benefit. So Jogjakarta, so shifting to Indonesia, releases have been undertaken as small pilots in these locations, and I believe a number of people went to visit one of these locations yesterday. These 4 pilots have been successful. 2 of them were done with egg releases, with containers, and the other 2 were done with adult releases. And we're getting ready at the moment, and that's what this exercise, this workshop is about, in preparation for doing a large-scale trial. I've been showing you observational data about no dengue transmission, but what we want is a randomised controlled trial that will give us the highest quality evidence for the impact of Wolbachia. And we would like to see that happen here, led by the Indonesian team. And this is just it's very good for an Australian audience to look at this and get an immediate appreciation for the increase in population density here compared to Australia. So just some results. And people from the Indonesian team can correct me if I get any of this wrong, or if Wasido, I can see you at the back there, if I'm not making sense here, you can jump in. But this is looking at 2 of these sites, showing the Wolbachia frequency successfully establishing over 2014, 2015. And these are dengue cases that have been reported in this area. Again, we need to be very careful about thinking about just dengue cases by themselves, because, you know, if you go and work in another part of the city during the day, you could get dengue there and then bring it back and be reported here. So this key feature is about local transmission and about clustering of cases, and we're not seeing any geographic clustering of the cases that we're seeing in these sites. And similarly, these are the 2 other sites where Wolbachia has been introduced successfully, and again, looking at dengue cases. You can see an obvious reduction there, but I think that the issue here is, again, around clustering. So, again, the data is pointing in a very good direction. This is just some data showing surrounding the release areas what's been happening, and so we've been monitoring off-site to see whether, which is the red and green, to see whether Wolbachia is moving out of the release areas, and we're not seeing that happening. Interestingly, this is an exclusion area in blue. So an exclusion area means that the community in this area did not want to see releases take place, and as I indicated to you before, we take that very seriously if the community doesn't want it, and so we didn't release in that community, but we monitored it in it. And in monitoring the community, we've just been seeing if there has been any flow in of Wolbachia from the surrounding areas. And there has been a little coming in, probably from mosquitoes moving into the area, but there's been no establishment of Wolbachia. Wolbachia is only establishing in the areas where we're releasing it. And currently at the moment, this is this public campaign. I don't know whether people who are from Jogja have seen it. I think it's quite interesting to see the creative ideas different project sites come up with for raising awareness, and this one is right up there with some of the more crazy ones that I've seen. Spectie's still in the room. I shouldn't say that in front of her, but I think it's really quite effective. And so, you know, our public awareness is very localised, and the groups that are leading the projects locally make the decisions on how best they think the public should be engaged. And this is what's going on, a public campaign right at the moment here in Jogjakarta. And this slide is just showing some of the stakeholder engagement that's been happening here at the project. So not only are we communicating with the community, but also with a broad range of stakeholders. And that's happening not just within Indonesia, but globally as well. I'd like to move to Vietnam and show you quickly what this area looks like. The site we've been working in. Whoops. This is a site we've been working in Vietnam now since the very beginning of the project. It's a small fishing village near the city of Nha Trang in central Vietnam. The city on the mainland has a population of around 500,000 people, similar to Jogjakarta, and this fishing village around 3,000 people. This is just showing you the city across on the mainland. So we've, in 2014-15, successfully released WML mosquitoes onto the island and of around 3,000 inhabitants. And again, we can look at some observational data on dengue. And here is the island, Tri Nguyen. It's 1 kilometre away from this mainland district. And this mainland district, we've compared the cases here to here from previous years. Here's the mainland, the dengue cases occurring, and then here's on the island, and we're seeing again a reduction in dengue with the release of the Wolbachia. We're also working in Colombia. This is, it's called Paris, but it's not Paris, France. It's Paris next to Medellin in Colombia, and it's not as nice. But it's a fairly small area that has around 40,000 people living in it, and about 6,000 residences. And we've been releasing mosquito into this community during that time period successfully. So the Wolbachia has now established into this neighbourhood of Colombia. Just let this run out so you get a sense of what it's like there. The housing is constructed. It's quite high density of people living there. And this is, you can see that it's quite steep. And this area is a big valley. And in the valley is the city of Medellin, made famous by the Medellin drug cartel of the 1980s, led by Pablo Escobar. And we're in the process now, based on the Zika emergency, this is a satellite view of the city running down this valley. This is the area of Paris that we have just released into successfully. We've now started an expansion through this valley where our immediate expansion plan while we're building up new rearing facilities and so forth is the phase 1 extension out of our existing area to cover another 55,000 people. And then we're moving into this area of Bejo and into northeast Medellin, with where we'll be deploying over the next probably 18 months across about 1 million extra people. And then the deployment is being extended across the whole valley to another 1.5 million people over the next 2 to 3 years. This is not going to be an efficacy trial. This is a scaled up deployment, and we're wanting to use this deployment to be able to get a better sense of how to work at that scale level and how to reduce cost. We will still monitor the impact, but it won't be done in an efficacy trial. We'll be using the existing surveillance system and enhanced surveillance in a number of Sentinel sites. And we'll be running with an independent external advisory evaluation group to look at the impact as a recommendation from WHO. And then this is in Brazil, where we also work. This is one of our first release sites called Tubiacanga. It's in the city of Rio de Janeiro, a huge city in Brazil. And we also have another location in Jurujuba. So similar to the Colombia in housing style and construction, and population density is a little lower. So just to give you the plan here, this is currently our release site, this little green dot where we've successfully released now. We're in the process of doing immediate expansion into this purple area, which has started now. This is again in response to Zika. And then our plan for phase 2 of around 500,000 people over the next year or so will include much of this area known as Niterói. And then phase 3 will be moving into high-risk barrios in the main part of Rio de Janeiro. And again, using a similar system for monitoring impact as in Colombia. So while this efficacy trial is running in Jogja, we hope, we'll have large-scale deployments happening in South America, and these are part of the expansion that's going on in response to Zika. So that's all I wanted to say as background. I've covered a lot of ground from safety to current deployments. If anybody has any questions of me, I'd be really happy to be able to explain what I know of what the current state of understanding of different aspects of the biology. No? Yes.